first of all, thank you, Gopal sir, for the opportunity and for a very kind introduction, Ashwin. I am Hitarthi Vyas, as you already might know, and today I'll be talking a little about clock-controlled cardiovascular health and the regulatory role of microRNA in that. So this is a part of work that I did in my PhD days. And then in a brief overview, if I can say, I'm just going to talk about how cardiovascular health is impacted by circadian rhythms. It has been known, but here we are trying to pin down the molecules that actually connect the two, and these molecules are microRNAs. So to begin with, yeah. So this story goes back a couple of hundreds of years. This was the first man, Jean Jacques de Marian. He observed chronodisruption in about 1700s. As you can see over here, the first observation was made in 1729. His observation was based on a plant, a heliotropic family of a plant. These plants are typically such that they open their leaves in the morning and then they close their leaves at night. But what he did was he shut a plant into a dark place, and even then, in the morning, when the morning time came, the leaves opened. So it made him very curious to further give a reason that it was not the sunlight that was stimulating the opening and closing of the plant, but the internal clock of the organism. So this was the first observation that was made of the circadian rhythms being present in an organism. And yeah, and then the first Nobel Prize, or the first major breakthrough that we made in chronobiology, was in 2017. So it took almost a good 300 years for us to make a major breakthrough as to what exactly happens in chronobiology and how is it impacting our human and all the rest of the organisms. Like, most of the organisms have their circadian rhythms trained in to the 24-hour daylight cycle. So, to give you a brief introduction of what circadian rhythms are, as you can see very clearly on the left side of your screen, the word itself is self explanatory. Circa, as in around, and the diam means a day. So, whatever goes around the day is known as circadian. That's what the word means. And our entire life is entrained within 24 hours. The sleep wake cycle is compatible to the 24-hour daylight cycle that goes around on the earth, of course. So here is a very decent picture that I use in a lot of my talks is the circadian rhythms and their physiological intervention. So if you start, typically all of us wakes up in the morning, and that's where melatonin secretion stops. Now melatonin is a biomolecule which is responsible to make us fall asleep. So when the secretion of that stops, we wake up. Slowly and steadily, our testosterone or the rest of the hormones, the cortisol, everything goes up, which means we are wide and awake. And then, as you can see over here, I would like to draw one attention, which is very critical observation that a lot of people made in with the athletes. If you see a lot of sports events, especially the 100-meter race and stuff, they happen in the noon because scientists have identified that this is the window where your body performs at its best. So a lot of these events are organized at about noon time. And then you start eating and you're fully awake because your body is entrained, like all your hormones which you need, for a complete activity, are produced during this time. Your heart rate are fine, your blood pressure goes up, you're in a completely alert mode. And when the evening sets down, again at about here, your melatonin secretion starts going up, which means you'll start to feel dizzy and you sleep off at night. So this is how a normal human body reacts to a 24-hour daylight cycle. But it's not always that simple, right? There are a lot of disturbances that goes on, and especially with current lifestyle, uh, it's not that simple. So I in the on the left side of your screen, if you see, these are a couple of reasons that disrupts your circadian rhythms, or also known as chronodisruption, like chronobiology and chronodisruption. When your circadian rhythms are disrupted or misaligned, that is the word which we typically use. So one of the major reason is shift work. A lot of doctors, policemen, IT engineers, call centers, all of these guys work late at night, or in fact work through the night, and go home, sleep home in the morning. They are doing completely opposite of how nature has created a human body to work on. Then, of course, the people who are transcontinental travelers, if you're coming, say, from US to India or India to US, one or 2 days you'll at least be awake at night and feel dizzy in the day, which is very common. And this the most common terms which you would have heard is jet lag. You must have heard that people like, oh, I'm jet lagged, I'm sleepy. So this is because your body is habituated, all your hormones and everything is secreted based on the 24-hour daylight cycle. Now if you move from US to India, the day and night completely shifts, but your body doesn't know that yet. So that is one of the major reason. Another, a freshly coined term is social jet lag. This is new. This is when, so Friday night, Saturday, you have a weekend off, you go out, say party, you come home late and sleep at about 2 a.m. at the night and wake up at 11 a.m. in the morning, which is not your usual week hours. If you're, say, habituated to sleep at about 11 at night and wake up at about 7 in the morning for 5 days a week, and for the rest 2 days you go haywire, that is what creates social jet lag. Again, the artificial lights at night, you being on the phone or surfing through the net, everything that disrupts your regular routine cycle is responsible for you to develop a chronodisruptive symptoms. And one of the major reasons, which is not there in India, but in US especially, is daylight saving time. That is also, it's a difference of a 1 hour difference what is done, but still that also has a lot of impact on the circadian rhythms of the body. So now, if you pay your attention to the right side of the screen, here is a human figure. We'll start from top to bottom of what changes when chronodisruption takes place, or when your circadian rhythms are goes haywire. First, you're thinking that's not very clear. You're not able to pay attention in the class. A lot of you must have experienced that if you're awake late at night and if you have an early morning lecture, you go to the class, your teacher is speaking, but you're so dizzy to grasp anything in your mind. So that is one of the classic symptom of chronodisruption. Your mood changes, your hunger changes, you gain weight, there is lower sex drive, your reproduction, or also your reproductive organs also takes a hit. And that is one of the major reason why infertility is getting such a big issue these days. Your immunity weakens. So in short, entire physiology is entrained to the circadian rhythms that are entrained to the daylight cycle. If you don't follow the pattern, your physiology takes a hit. Now, this is one of the very interesting diagram that I loved, so I put it over here. If you see on the x-axis, as the axis progresses, that is the age. As you progress from baby to teenager to an old adult, and on the y-axis is the disease severity of how the diseases progress. So I would like to draw your attention to a couple of things which you must have with the coins that are, or sorry, the terms that are very common these days. First is obesity, which is like a major uh, pandemic across the globe. People are getting obese. Then anxiety or panic attacks or depression. Like when we were kids, we never heard of these terms so commonly, that, oh, I'm having a panic attack, or I'm getting depressed. These terms have gotten more common these days. A lot of people you must be hearing here and there. So these are some of the major outcomes that are seen. Narrowing down again, over here is the heart disease. This is also getting very common. Especially after COVID, that's a different reason altogether. But heart diseases and heart conditions are also getting very common these days, and I think it has a lot to do with how your circadian rhythms are taking a toll because of all the social media and the phones and everything. So in here, if you see on the left side of your screen, this is a very, very superficial, a broader picture of how the circadian rhythms work. So I'll walk you through this, and this will help you in the later part of the presentation. So, for the light enters into your retina, and that sensitizes the suprachiasmatic nuclei. So, this is a set of neurons in your brain that gets sensitized, and that further triggers your physiology and your behavior, both. So, this is a broader picture. Now, if you move to the right side of your screen, here are the common molecular mechanisms that goes on. So now, as you know, that it's a cycle, daylight cycle, so it operates in a loop. So even these molecules have a feedback loop system. They work in a negative feedback loop. So the core molecules are clock and bmol and cert, and the peripheral clock molecules are per, cry, reverb, roar, etc. So what happens is when BMAL and clock sets in, it sets on the E box, and that leads to transcription of all these peripheral clock genes. When the level of these genes increase, the clock and BMAL are dissociated, and the transcription stops. So basically, it operates in a feedback loop. So from this image, I would want you to remember SERT and clock. That is the molecule that we are going to see further through the presentation. So after that, if we can zoom in, we saw how it works at the physiological level and at the pathological level. So now I would want to draw your attention to a little bit narrower to the cardiovascular operation, because that's where we are going with this. So if you can see over here on the top left corner is the heart rate. So this experiment was done in 1989. This was a couple of the first observations that were made linking cardiovascular system and chronobiology. So this here, the blue bars that you see are basically the heart rate through different hours of the day. So this starts at the midnight. It goes up at about 7, 8, which is typically where a lot of people wake up. It stays high up throughout the day, and it starts to set in towards the end of the day. This is also absolutely complemented by the systemic blood pressure. If you can see, this also has a similar bell pattern through the course of 24-hour time of the day. So these are the physiological observations that were made. Now, when you come to the right side of your screen, here, this is a blood vessel. Again, I've chosen this because we'll be talking about atherosclerosis, which typically happens in the blood vessel. So these are the couple of events which are very significant through the course of atherogenic development. So when your circadian rhythm goes haywire, the inflammation goes up, the oxidative stress goes up, your hormonal imbalance is a very significant part. Your estrogen, testosterone goes down, your angiotensin, aldosterone, everything goes up, and autophagy, of course, goes down. So basically, a conglomeration of all these events takes place that initiates and progresses atherosclerosis. So till now we have seen what chronobiology is, or what chronodisruption is, and what are the major facets of human physiology that it impacts. So now here in the screen, you can see we have a cell nucleus. This is a central dogma, right? So you have your DNA, your mRNA that is getting transcribed, that is further getting translated into a protein. But I would want to draw your attention to these teeny molecules, very small 20 to 22 nucleotide repeats, which are known as microRNAs. So these are 22 nucleotide, typically RNA, non-coding RNA molecules that are associated with the RISC complex. Now, RISC complex is a protein, as you can see in this image uh, right here. Yeah. So, what microRNA does is two things. MicroRNA can first bind to the mRNA and degrade the mRNA completely. So your cell is given signal to your nucleus that produce this mRNA. The nucleus is producing the mRNA, but as it goes out, your microRNA binds to it and either degrades it or it impedes the translational process. As you can see on the right bottom screen, the translational machinery is not able to sit on the mRNA, and hence no protein is being formed. So I think this is a very interesting fine tuning that happens in at the cellular level, that controls a lot of function. So then I became interested to see what these molecules have to do with chronodisruption and cardiovascular disorder, and especially atherosclerosis. So till now, whatever we talked, it was a cause-consequence relationship. That chronodisruption takes place or circadian rhythm are inset. These are the physiological processes that happen, but there is a huge gap that exists when it comes to what are the biomolecules or what are the systems in place that regulate these interaction or cause consequence relationship. So this study is somewhat an attempt to connect the link between the two. So I'll give you a brief introduction of what atherosclerosis is. So it's a very complex pathophysiological process that goes on. So I have divided the screen in one, two, and three parts. So I'll walk you very briefly through the disease pathology over here. So now this is the endothelial cell lining. This is your vessel when I'm talking about, which is the innermost lining, the endothelial lining, and the blood flows over here. So whenever there is any stress, be it hyperlipidemia, hypertension, diabetes, or turbulence in the blood flow, especially when you have the vessels that are arching like this, the blood flow and the pressure changes to at the arch. So a lot of atherosclerotic plaque tend to develop in these arches. So all these tensions cumulatively triggers the endothelial lining, wherein they pop out the adhesion molecules. As you can see over here, if my pointer is visible, the blue, green, red, these are the adhesion molecules. And when they are expressed, what happens is your endothelial cell is saying that, okay, I'm stressed. So what happens? But obvious, there are
पॉइंटर
monocytes.
नहीं हो तो क्लिक कर लो पॉइंटर को, तो अच्छा से दिखा पाएंगे. पॉइंटर का जो है ना थोड़ा मोटा होगा,
Visible hai, sir?
तो नहीं पॉइंटर वाला अलग होगा. आप देखो. हाँ
Yeah.
हाँ हाँ, अब दिख रहा है. अब थोड़ा सा.
Yeah.
यस.
So what happens is there are monocytes freely circulating. Sorry about that. Freely circulating in the blood vessel. These adhesion molecules capture those monocytes and they are internalized. Like here. So once the monocytes are internalized and there is excessive lipid content that is flowing through, monocytes start to eat all those lipid and it becomes very bulky. This is where it has been converted to macrophages and foam cell eventually. After a point, it keeps on eating the LDL, but it becomes very bulky and it breaks. Here we have entered into the second half. And in the third half, eventually all these debris, LDL, everything together starts building up the necrotic core. Now, when I talk through this process, in complementary to that, simultaneously your ROS is going up, your inflammation is going up, a lot of signaling, a lot of immune signaling are being sent out, and it's like a loop process. More and more monocytes are recruited and the foam cell formation starts. So as you can see over here, this is the development of fatty streak. At this point, your plaque is starting to build up, but it is still in a smaller size. Once it reaches to this size where it becomes very bulky, the media layer of the your vessel starts to send out your smooth muscle cells to cover up the plaque, just to increase the stability so that it doesn't rupture. But after a point of time, if the plaque ruptures, the debris and the lipid core and everything, if it gets stuck into a small arteriole, there is no blood flow further to that organ wherever the debris or the plaque is getting stuck, and so there is no oxygen, and this is when the hemorrhages are caused. So it's a pretty serious pathology that happens. But for this talk, or for my study, I have focused just on the initial part of what happens, because I wanted to focus how circadian rhythms initiate and progress the the atherosclerotic pathology. So I will dive into the data now. I know it might be a little data heavy, but I'll talk more about it conceptually. So I used first the couple of softwares, the algorithms that showed me how microRNAs uh, bind to the 3' UTR of clock gene. So here, whatever you are seeing is a couple of microRNAs that were spat out of the system for humans and for mice, and we found about 83 of them to be common. So I further processed those 83 again through the softwares and the PubMed screening and the literature that was published, and eventually we narrowed down to MIR34A5P, and kind of skipping a little bit just to compact the story and deliver it in interest of time. So MIR34A5P was the microRNA that we thought must be the potential link between the two. So to begin with, we screened for ME34A sequence that was pretty conserved across the vertebrates, and in humans, mice, and rat, those are the models that we were going to use for the experimentation purposes. So here you can see that there were seed locations, or basically the site where microRNA MIR34A would bind to the 3' UTR of clock gene and stop its expression. This is again one of this was again I did for the confirmation. As you can see here, these are the pathological conditions that were potentially regulated by ME34A5P, and this is the gene ontology that was perturbed through this microRNA. Now, as you can see over here, ME34A5P binds to a lot of microRNA. Also, I'll take a moment here to talk about one unique property of microRNA that made me very interested to go and work on it. Was one microRNA can bind to a lot of different mRNAs. So as the first graph here that you see is what I did was I saw how many genes are actually shown to have implications in cardiovascular disorder. And out of those, let's say they were 100 genes. So out of those, what percentage of genes were actually had a complementary seed sequence to ME34A5P? So from that, we deduced that about 40% of the genes functional in cardiovascular disorders had a complementary seed sequence to ME34A5P. So this was a pretty solid start that gave us a good confidence that ME34A5P can be a potential candidate to further be explored. So then we went to the endothelial cells. The cell line that I've used is HUVEC, human umbilical vein cord cells. Now, when you have a cells in a dish, uh, they do not have a sync circadian rhythm like our body does, because we are one entity, but the cell in the petri dish may not. So first you have to serum synchronize them so that all the clock are at place. And then I wanted to increase ME34A5P expression. So I gave LPS to the cells, seeing that the clock and the SERT, the molecules of interest, are not getting disturbed, just your microRNA is getting high. So in those conditions, I checked that when MIR34A5P levels were increased, the clock and cert levels were going down. So this kind of confirmed what we saw earlier in the net form spat out by the algorithm, that MIR34A5P actually bound to the 3' UTR of clock gene and stopped its protein formation. Here at the bottom is the ICC, and here's the quantification telling the same thing. So we have looked at it at the mRNA level, protein level, through Western and ICC, just the confirmation through different techniques. Uh, Now that being said, we wanted to actually confirm. So what we did was we developed a plasmid with wild type clock 3' UTR, and one was the mutant one. So here is how a plasmid looked. I'll just walk you through the 3' UTR of the clock. One was normal and one was mutated, and that had a Lucifer reporter gene after it. So if the MIR34A5P bound to the 3' UTR of the clock gene, the Lucifer reporter gene would be expressed in a lower, would show a lower expression as compared to the mutant one, and that is exactly what we saw over here. If you can see the red bar, the luciferase activity is less over here, and which was almost compared to normal in the mutant clock. So this was the final confirmation experiment that MU34A5P actually impedes clock gene expression in the body. So now when I talk about circadian rhythms, I wanted to see what happens at the 24-hour time scale. And here, as you can see, Me34 as the Me34A level goes down, clock and cert are high, and vis-a-vis, which happens in a cyclic manner. So this is the mRNA and the protein over here. So that was again a confirmation of they express in the cyclicity. So after looking everything in a cell based manner, I further went to the actually animal models, and here we used mice, C57BL6J mice, and we subjected them to photoperiodic manipulation. So a control group was like this. The yellow lines or the streaks that you see over here were the daytime, wherein the lights were on, and the blue lines represent the nighttime, and the lights were off. But in a chronodisruption condition, what we did was we did a phase advance and phase delay of about 6 hours twice a week. And this went on for 18 weeks. So we developed a chronodisruptive model, uh, which would serve for our further experiment purpose that I'll talk about. So just to confirm that the model was fine, I did a couple of clock control genes that we talked about earlier, clock, bmol, per, cry, etc. And the levels went down, confirming that the chronodisruption has actually occurred in the mice. Okay, and when I talk about mice, I'm talking about the thoracic aorta. I isolated the thoracic aorta, and all these gene and the protein expression that you'll be seeing is of the aorta. Now, here again, a second validation through the protein and immunohistochemistry showing the same thing, that in chronodisruptive mice, these molecules, the clock bmol cert would go, the expression would go down. So it was great. We confirmed that Me34A5P binds to clock, the expression goes down. But then the question was, what happens, what makes Me34A expression go up in chronodisruptive condition? As you see over here, it's almost twice the expression of in as compared to the control. So then I turned my head to the epigenetic model. So this is very common when your CpG island is typically methylated. It doesn't express that much. So the demethylation, as you can see over here, if the methylation goes down, the transcription increases. So we took a couple of primers through the landscape of Me34A5P to see the methylation and unmethylation that is happening during the phase of chronodisruption. And this, in fact, was very promising. The methylation would go down in the chronodisruptive condition. And again, to complement that, we also did the unmethylated set. Uh, so it was amazing how epigenetics of the MIR34A itself were changing, leading to increase of its expression in the thoracic aorta. So now the entire story as it comes to the atherosclerosis. So I wanted to check in this chronodisruptive mice, what is the status of atherosclerosis per se. So here, the first gold standard parameter that you do is assess the lipid profile. And this is the atherogenic index of plasma, which is, there is basically a formula wherein you put in the value of your HDL, LDL, TG, and it spits out the atherogenic index of plasma. And likewise, the cardiac risk ratio. As the names are self-explanatory, it shows the atherogenic index and a CRR, which were very significantly high in chronodisruptive condition. So I further went to check what is the status of the thoracic aorta of this animal. And as you can see over here, there was intimal media thickening. The collagen to elastin ratio was also off, and there were a lot of elastin strand breaks, making like proving that the thoracic aorta itself had taken a good hit because of chronodisruption. So for this part of the study, the what we found was the mechanistically how 34A regulates with certain clock and how it impedes the circadian system. After this, I wanted to go to next part of the study and make it more focused. So I used two molecules. One was CO, that is carbon monoxide. So during this course, I came upon some of the papers that showed that carbon monoxide had circadian implications, and it could also regulate the clock gene expression. So I went to a more focused model, that is flag dolly rats. This is a more convenient model to study atherosclerosis, and my lab actually had used this for a while back then to work on atherosclerosis. So I developed an atherogenic model by giving vitamin D and atherogenic diet to the rats. And as you can see, so these are the thoracic aorta that I had isolated from the three groups. When I said a brief introduction about carbon monoxide, so I used carbon monoxide releasing molecule A1, that is COM-A1, to suppress MIR34A5P and to also see its regulation on circadian rhythm. So that was the molecule which would regulate MIR34A5P. That I'll show you as we go further through the presentation. So basically, over here we have three groups: the control, the estrogen, the AT group. As you can see, the lesions, if it is clear, like if you can see through the presentation. Otherwise, believe me, there were lesions in the thoracic aorta. This is where the plaque development would start, and the COM-A1 group. Again, so here I would want to walk you through the same slides, the histomorphological changes through the thoracic aorta of the rat. As you can see, now because this is a more relevant model for atherosclerosis, you would see a very pronounced intimal media thickening over here, which was brought back to almost comparable to control in the COM group. And this is just a negative control for COM. Again, likewise, elastin, here you can see a significant strand breaks and the picrocerous red stains for collagen, showing the similar things. So as I talked about earlier, these are the CRP, that is inflammation marker, the atherogenic index of plasma, and cardiac risk ratio that we did to confirm in the SD rats, and these are the lipid profile. This is getting a little data heavy, so I'm kind of browsing it through. So again, we came back. What happens to me 34A5P? Now here forward, A lot of data has three things, that is thoracic aorta of rat, the UVEC, the cell lines, and MDMs, which are the monocytes that I talked about earlier. So a lot of things I've done in all the three models just to gauge what exactly happens at the systemic level and at the cellular level. So MIR34A5P increases in all the disease condition when you trigger, but when you give COM-A1, it suppresses. As I was just talking to you earlier, MIR34A5P levels goes down with COM-A1. So henceforward, whenever I say COM-A group or COM-A1 group, what I essentially would mean is lower MIR34A5P. So then I became interested to see what exactly would regulate the expression of MIR34A5P. How would COM make it go down in atherogenic condition? So typically there are three routes through which you go. One is transcription factor, inhibitor, and epigenetic modification. So I went about all of them turn by turn. So when I was working on it, this is the broader picture that was known, that P53 NF kappa B were the transcription factors of MIR34A5P, and ZEB, SNI, and STAT3 were the inhibitors. So I went about studying all those three at the protein levels and the mRNA levels in all the three models, the aorta, UVEC, and MDMs. Um, in all the three models about that. So here P53, if you can see over here is the P53 mRNA and NF kappa B, where NF kappa B acts pretty significantly when you add COM-A1. The levels of NF kappa B goes down, and so happens for P53. So the transcription factors are going down when you give COM-A1. Now I further moved to what happens to transcription inhibitors. Again, the same three models, and these are the inhibitors that I just talked about earlier. And all the inhibitors were actually pretty much going high across the model. So, which was very significant that COM-A1 was acting through transcription factors and inhibitors. But then, as what the epigenetic modification, as you saw earlier, that I had identified in the mice. So I wanted to check the same thing, what happens to the methylation pattern, because it turned out that the that portion of MIR34A5P site is pretty sensitive to methylation and demethylation. And to our surprise, we got exactly same thing over here. The methylation would go up, and hence we were seeing lowered MIR34A5P expression on giving COM-A1. There was one more thing which we then thought of doing. What would happen to the physical proximity? Like, would CO interfere or associate physically to the transcription factors and inhibit their binding to the DNA or not? So we further did we our collaborators did that for us, and here, as you can see, we get a pretty good negative glide score for P53. CO would bind in the DNA binding domain, which was again very interesting to me, that it was not only regulating through transcriptional factors, but also through the physical proximity binding to the DNA binding domain, and hence that was showing such a significant effect on MIR34A5P. So after that, as you might remember, the first graph that I showed you earlier that talked about clock and SERT1. So we saw what happens to clock and MIR34A5P. Now in this half, I would want to talk about a little about what happens with SERT1. So MIR34A pretty much binds to SERT1. It was known by that time. So we straight went ahead and did just a little confirmation experiment. Here, the IB graph that you see is the antagomide, which typically does not allow MIR34A to bind, and hence you would see the SERT expression over here, which went down in the atherogenic conditions. And again, when you give CO or the COM-A1, the SERT expression went up. And when you do not give COM-A1 but still give the miR34A5P integromere, the SIRT1 goes down. So this is just a confirmation that miR34A5P regulates SIRT1 expression. Again, this was done through all the three models, the HUVEC, MDMs, and potent or important biomolecule in regulating, especially mitochondrial biogenesis. Actually, SIRT1 is at a position where it regulates a lot of different processes, but especially mitochondrial biogenesis through PGC1 alpha activation. So I went down that line to check what happens to SIRT1 PGC1 alpha axis, because in atherosclerosis, mitochondria is very important. It takes ROS generation and inflammation and stuff is a huge impact on the pathological initiation and progression. So I wanted to check what happens down that route. So this is just the Western and mRNA expression of PGC1 alpha that goes down in atherogenic conditions, as you would expect, and increase in miR34A5P lowered condition or COM-A1 complementation. Again, HUVEC, MDM, and IOTA. Uh, with PGC1 alpha, I also checked the downstream mRNA levels that were NRF1, DRP1, TFAM, all again showing mitochondrial biogenesis, pointing towards mitochondrial biogenesis, that the levels were increased. So after looking into the gene expression levels, I went to check what happens to mitochondria, like visually. So I used the stain mitotracker red over here. The more red you see is the more number of mitochondria. It stains the mitochondria. So it is very evident that when you develop atherogenic group, the mitochondria number goes down, but in presence of COM-A1 or lowered miR34A5P, the mitochondria increases. Uh, I did the same confirmation again through mitochondrial DNA. I checked mitochondrial DNA because if there are obviously more mitochondria present, there should be more DNA. So this is just a double verification of the same thing, and which showed a pretty complementing results, as you would expect, across HUVEC, MDM, and IOTA. So it is not just enough that you have more mitochondria. You should also have a good or a healthy mitochondria. So I checked mitochondrial membrane potential through JC1 staining, and as the figure shows, the if you have a higher membrane potential, you would see red in color, and the lower were shown by is depicted by green color of the stain. So here are the representative images that I took. And of course, in the atherogenic condition, you would see more green mitochondria, and that's what we saw for both HUVECs and MDMs, and that were restored on giving COM-A1. I would kind of skip this. This is again talking about the redox status, regulating the ROS of the environment in HUVEC and MDMs, which was restored in just an interest of time. After looking at mitochondria and mitochondrial redox status, I also looked at the cellular redox status. Now, this is done by a different stain, DCFDA. And here, as you can see, more green again is more ROS, which was restored when you had lowered miR34A5P and COM-A1. So after you have more mitochondria, you have healthy mitochondria, you need your mitochondria to function, right? So I looked, and mitochondrial function is, of course, the major function is, of course, ATP production. So I checked ATP production again in HUVEC, MDM, and IOTA, and I got similar complementing results, that on giving COM-A1, the ATP production was restored. Now, one of the interesting things that I would want you to see, draw your attention to, when you work in a cellular system, the ATP production has gone down, but not as significantly as you see in the actual in vivo system. So when you have a complex pathology, what an animal would show, not all the cells would show. So this was very interesting that I thought through the course of time. And finally, this is the mitochondrial functional assessment assay that is done by the seahorse, wherein you measure the oxygen consumption rate, known as OCR, abbreviated as OCR, and extracellular acidification rate, known as ECAR. This basically spits out how your mitochondria is doing exactly. So here you would get the readout would be in a basal respiratory complex, the maximal respiration rate, your ATP production, just showing how your mitochondria is acting. Now, this is again a little interesting. What I did here was we gave MIR34A5P antagomer, that is depicted. Sorry, I should have had key over here, but that is depicted in a purple line. So when you look at THP1 or MDMs, named earlier, the green line, which is your COM-A1, and your Mi34A5P antagomer, the level of expression is exactly the same, showing that whatever restoration is happening after giving COM-A1 is through is to decreased Mi34A5P in MDMs. But this little space you see between green and the purple line shows that there are some mechanisms active apart from MIR34A5P that are driven by COM-A1 to protect the mitochondrial status. Here, this talks about the glycolysis and the glycolytic capacity, again, showing a similar result of mitochondrial function. And I think I'm almost at the time. So, yeah, this is the brief summary. So, when you give, this is a pictorial representation of what I did. When you give COM-A1, it regulates the transcription factors, epigenetics, and the transcription inhibitors that further regulates MIR34A5P, SIRT1 expression, and the mitochondrial biogenesis, function, and status. This was one more part that we did not touch in this talk, but I did it, so it's there in the figure. And with that, I would like to acknowledge my previous research group, my present research group, and our collaborators, and all the fellowships that I have received through the time. Thank you. If you have any, let's see.